My name is Anna Cunane, and my undergraduate research is on engineering fluorescent fusion protein with novel electron uptake gene in Shewanella oneidensis. So what is electron uptake? Electron uptake and electron deposition is how our bacteria, Shewanella, eats and breathes. Human beings will breathe oxygen, but Shewanella will breathe insoluble materials and deposit electrons from their food onto a poised electrode or mineral. This will reduce the electrode or mineral, and we would call this mineral reduction. Shewanella can also take in electrons from an electrode by oxidizing it. This process of electron exchange between the microbe and mineral or electrode is termed extracellular electron transfer. Mineral reduction has been fairly well characterized, but the reverse of this process, mineral oxidation, has yet to be completely understood and defined. Previously, we did a genome-wide screen of non-essential genes, searching for phenotypes of non-electron uptake. From there, we identified 5 genes encoding for proteins that were possibly involved in this electron uptake pathway. A few of these proteins were thought to be associated with the cytoplasmic membrane, and this figure displays our hypothetical pathway for how the electron uptake process is occurring within the cell. One of these genes, termed SO3662, is thought to produce a ferredoxin protein or an iron-sulfur protein that is thought to be involved in extracellular electron uptake. My job was trying to determine the localization of this protein within the cell, and we hypothesized that this protein is located in the cytoplasmic membrane. To help us determine protein localization, we used an E. coli plasmid containing a cyan fluorescent protein, which will help us to locate SO3662 after we turn the gene on. The plasmid was cut on the front and back end of the cyan fluorescent protein, and the gene was inserted at restriction enzyme sites on both sides. The plasmid was then transferred from E. coli into Shewanella via conjugation. We then induced expression of the gene for different periods of time. After we induced gene expression, we were then able to use microscopy techniques to try and visualize protein localization. The cyan fluorescent protein microscopy image shows us the location of our protein indicated by the blue glow around the outside of the cell. We also used a lipid counterstain indicated by the TRIP stain, which stained both membranes of the cell so we could try and distinguish which membrane the protein is in. We then overlaid the counterstain and the CFP gene expression to get a better understanding of where the protein was within the cell. These images would indicate that the protein is found in the outer regions of the cell, but further research must be done to confirm if it is localized in either the outer membrane or the cytoplasmic membrane. Our next step would be to confirm protein function and possibly manipulate the electron uptake process. So how can we apply this research? If we are able to fully characterize this, the manipulation of Shewanella's electron uptake pathway will have many biotech implications, and it will also be useful in electrosynthesis research. Currently, there is research being done on using Shewanella in microbial fuel cells, and upscaling of this process would eventually lead us to new clean energy sources. I would like to thank the Air Force for funding our research, and Roe Labs for letting me come on and do research with them, and a special thanks to Josh Sackett.